Hi everyone, greetings and welcome back to my educational channel on biology. In this video, we're going to discuss Form 4 Chapter 9 Biology KSSM syllabus, subtopic 9.1 Digestive System and 9.2 Digestion. Now, food is something that's important to us, and from the moment we eat a burger or a plate of char kway teow or roti canai, our fantastic digestive systems kick into action and begin the long process of digestion, secreting enzymes and squeezing the food along the way to help extract simple nutrients for our bodies to absorb and use. So let's find out how this digestion occurs, Let's embark on a journey together to find out how the digestive system extracts valuable nutrients from food for the use of the whole body. The learning standards for today's lesson are as follows. 9.1 Digestive System. After this lesson, we should be able to identify structures of the human respiratory system, including the digestive organs. 9.2 Digestion. Describe the two types of digestion, which are physical digestion and chemical digestion. Next, analyze the process and products of carbohydrate digestion that occurs in the mouth. Then, analyze the process and products of protein digestion that occurs in the stomach. And lastly, describe or analyze the digestion of carbohydrates, proteins, and lipids that occur in the small intestine. Our first topic here is types of digestion. So, when you eat a piece of sandwich, for example, during your breakfast, when you eat the sandwich, do you know what happens to that sandwich in the body? So when we eat a sandwich, the process of digestion occurs and all our digestive organs begin to function to break down that piece of sandwich to form simpler molecules that are dissolved for easy absorption into the body, so that the body can use these nutrients for its needs, like for growth and for cellular respiration. So digestion is defined as the process that breaks down the large and complex pieces of food into simpler and smaller pieces that can be dissolved and be absorbed, for easy absorption. Dissolved for easy absorption. For example, when you eat a piece of bread, it is large, so we chew it and it becomes forms the smaller pieces of bread. On top of that, starch is found in the bread. It's the complex molecule. So it needs to be broken down to form simpler molecules such as glucose. The end product of all the digestion will be starch is broken down into maltose and then glucose, and then the glucose can be easily absorbed by the body. So digestion is made up of two parts: physical digestion and chemical digestion, the two types of digestion. Now, physical digestion is the mechanical breakdown of large food particles into smaller particles. So, mechanical breakdown. We use our teeth to chew on the food, right? So, physical digestion involves chewing in order to break down the food into smaller pieces. And then there's peristalsis, whereby there's contraction and relaxation of the muscles in the wall of the esophagus and other parts of the digestive system. So, physical digestion involves these processes: chewing and peristalsis. For example, chewing on a piece of bread. So when we eat a piece of bread, we chew it and it's broken down into smaller pieces. But this does not involve the breakdown of complex food particles yet. Now, in chemical digestion, then there's the breakdown of the complex molecules, in the food into simpler molecules. So chemical digestion is the decomposition or breakdown of complex molecules in the food to form simpler molecules. So chemical digestion occurs at a smaller level, molecular level, and it involves hydrolysis, reactions catalyzed by enzymes. So when enzymes are involved, then it is chemical digestion. Example, digestion of starch molecules by enzyme amylase to form maltose molecules. Okay, so when you eat the piece of bread, there are starch molecules in the bread, and starch is a complex polymer of glucose. So all these green shapes represent the glucose molecules. Now, the starch molecule needs to be broken down by amylase to form simpler molecules like the maltose, which is a disaccharide made up of only two molecules of glucose joined together, right? So in this way, the starch will be broken down to maltose, and then finally it will be broken down to glucose by another enzyme, and then the glucose can be absorbed by the body, right? So this is chemical digestion, whereby complex molecules like starch are broken down by enzymes through a hydrolysis process to form simpler molecules like maltose. Right, so the human digestive system consists of many organs that are involved in the process of digestion and breakdown of food. Starting with the mouth. The mouth is the site where the digestion of starch by amylase occurs. So salivary glands are found which produce the saliva that contains the amylase. Okay, so they are found around the mouth region. And then we have the pharynx, which is the intersection between the esophagus and the mouth. Here is at the back of the mouth. And then we have the epiglottis, which is a tiny flap that closes the trachea when we swallow so that the food doesn't go into the trachea. Next is the esophagus, which is a long tube-like structure that helps the food to move down to the stomach, right? So in the esophagus, the process that occurs is called peristalsis, to push the food down to the stomach. Next, the stomach is the second site for digestion, and this is where the digestion of proteins occur. Okay, and also there's production of hydrochloric acid in the stomach to kill the bacteria in the food. Next is the duodenum, which is the third site of digestion. So digestion of starch, polypeptides, and lipids occur in the duodenum. And the duodenum is connected to some accessory organs, okay, which help the duodenum in the process of digestion. Because the duodenum doesn't produce any digestive enzymes of its own, right? So it depends on accessory organs like the liver to produce bile, to break down to emulsify the fats or the lipids, right? And then there's the gallbladder to store the bile. And then there's the pancreas. All right, now pancreas is hidden behind the stomach. So let me bring it out to the front first. Now the pancreas secretes 3 enzymes that help in the digestion of starch, polypeptides, and lipids. Okay, whereas the liver secretes bile that is stored in the gallbladder, and the bile is to emulsify the lipids. Okay, next, the food goes on down to the next part of the small intestine, which is the jejunum, and then the ileum below. So duodenum, jejunum, which is not mentioned here, and ileum are all parts of the small intestine. Now the ileum is the 4th site of digestion of food. It digests the peptides, disaccharides, and lipids to finally form the end products of digestion. And then absorption occurs after that. Okay, absorption of nutrients. Now ileum is connected to the appendix and the appendix is the start of the large intestine. So the large intestine consists of the colon, which is an N-shaped structure here, and then we have the rectum here also. Actually, the first part here, there's one part called the cecum here. Never mind. Mainly is the colon. Okay, large intestine consists mainly of the colon, and then here we have the rectum and the anus. So for defecation of food. So here is a very useful table which I've drawn up to show all the different processes of digestion or hydrolysis involving all the different enzymes in the digestive system, starting from the mouth to the stomach, duodenum, and finally the ileum, right? So you can copy this down in your notebook, and it will be very useful to you when you do your revision. Okay, it summarizes everything that we're going to study here. So starting with the mouth, we're going to talk about digestion in the mouth. Now, we need to know the glands, the secretion involved, the enzyme involved, the substrate, and the product, right? So the gland in the mouth are the salivary glands, and they secrete saliva. Saliva has a pH of 6.5 to 7.5, which is almost neutral, okay? Neutral condition in the mouth. And the enzyme in the saliva is called amylase. Amylase hydrolyzes starch to form maltose. So notice here that I have put the hydrolysis process in the form of an equation, all right? Starch plus water produces maltose. So in the exam, if we're asked to write the word equation for the hydrolysis reaction, we can write it as starch plus water, spell out the water in full, produces maltose, right? And then on top of the arrow here, write the word amylase. Okay, so bring this word amylase to the top here. Okay, so this is a good summary of what we're going to discuss here. So let's talk about the digestion in the mouth, and that's the digestion of only one class of food, which are the carbohydrates. So the digestive process begins in the mouth once we start eating something. For example, you eat a sandwich for breakfast, right? At the moment you put into your mouth, the digestive process begins. The presence of food in the mouth will stimulate the secretion of saliva from salivary glands. So we humans have 3 major salivary glands, right? As seen in this picture. One, there are some that are under the tongue, and then one is at this position here, right? So the saliva will be secreted, and it contains an enzyme called salivary amylase that hydrolyzes starch to maltose. Okay, according to this equation here. So this equation is important for you to remember, especially when a question is asked like state or write down the equation, the word equation for the hydrolysis of starch, or write down the word equation for the hydrolysis of a substance like bread that occurs in the mouth. Okay, so the pH of the saliva is between pH 6.5 to 7.5, neutral condition, which is the optimum pH for salivary amylase to act at a high rate of reaction. Next, so the saliva will, as you are chewing the food, the saliva will help the food to form a bolus. A bolus is like a tiny ball or tiny bit of food, mass of food, right? Which has been chewed and mixed with saliva. And this makes it easier to be swallowed. Next, when you swallow the food, something moves in your neck here, and that is the epiglottis. The epiglottis is a tiny projection, a tiny flap that projects out above the trachea. So the position of our esophagus is that it is next to the trachea. The trachea is in front, and then the esophagus is at the back. Now, so we must make sure that the food doesn't go into the trachea, or else the person will choke. So the epiglottis this flap will close down when you swallow. Okay, try and swallow now. You can feel something moving downwards, and that is the epiglottis. So it will cover the trachea, the opening of the trachea, so that the food can go down into the right channel, which is the esophagus. Okay, so this will prevent choking. Now, so the epiglottis closes the trachea opening when we are swallowing to prevent food from entering the trachea and prevent the food from blocking the airflow into the trachea. Next, when the bolus is moving down the esophagus, it is moved by peristalsis. Now, this word has to be defined. Okay, it's important term and has been asked before in SPM. So peristalsis is the rhythmic contraction and relaxation of smooth muscles along the alimentary canal. Now, where are the smooth muscles found? The smooth muscles are found in the wall of the esophagus. So to move the food downwards, the smooth muscles must contract above the food. Above the food, the smooth muscles will contract to push the food downwards. Okay, so peristalsis, the function of peristalsis is to push the bolus through the esophagus until it enters the stomach. So here we have a picture showing peristalsis occurring. Right, let's say this is the bolus in the esophagus. Okay, so the smooth muscles above the bolus will contract to push the food downwards, but the smooth muscles where the bolus are will relax so that the food can be moved downwards. So once the food moves to position B here, from A to B, again the smooth muscles above the bolus will contract, and the smooth muscles in the region of the bolus and below it will relax to allow the bolus to move downwards. So this rhythmic contraction and then relaxation of the muscles is called peristalsis. Okay, and it occurs along the alimentary canal, even in the stomach and the small intestines, all right, and also the large intestine. Peristalsis occurs to push the food through the alimentary canal. So the story of the food continues. Now, number 2, the stomach. So imagine the bolus has arrived at the stomach, all right? The food has arrived at the stomach. Now, the stomach contains gastric glands that secrete gastric juice, which has a pH of 1.5 to 2.0 due to the presence of hydrochloric acid in the gastric juice. The enzyme contained in the gastric juice is pepsin. Pepsin hydrolyzes proteins to form polypeptides. This is the first stage in the digestion of proteins. Now, how about renin? The textbook, your new textbook, the KSSM textbook, does not mention renin, okay? But it is in the old syllabus and in the past, the old textbooks. So is renin present in the gastric juice? Now we'll discuss this in the later slides. So the function of renin is to convert the casinogen, which is soluble, into insoluble casein in the milk. So, but if you're asked to write one word equation or one reaction, describe one reaction that occurs in the stomach involving enzymes and hydrolysis, please mention pepsin instead. Please state it, pepsin. State pepsin, not renin, okay? Pepsin is your first choice because it's in the textbook and it is the prominent, it is the enzyme, the main enzyme there, okay? Now, how about hydrochloric acid? Hydrochloric acid kills the bacteria in the food and creates an acidic medium for the action of pepsin that needs an optimum pH of 1.5 to 2.0 for its fast action. Okay, so in the stomach, there's only the digestion of protein, no digestion of starch or lipids. Right, the surface of the stomach wall is lined with epithelial cells that have undergone adaptations in structure and function, and they form the gastric glands. So we'll take a look at the gastric glands in the next slide. Now, there are 3 types of cells found in a gastric gland. Okay, so remember, now chief cells, parietal cells, and mucous cells. So you can form an acronym. The chiefs are PA, Pa, Papa, and M, so Mama. Okay, the chiefs are Papa and Mama. The chiefs in the family are the father and the mother, right? Okay, so the chiefs are Papa, Papa stands for parietal cells and mama mucous cells. Okay, right. So chief cells secrete pepsinogen, right? And the function of this secretion, the pepsinogen, is that when pepsinogen is converted to its active form, pepsin, by hydrochloric acid, the pepsin will hydrolyze proteins in the food to form simpler substances called polypeptides, which are still polymers of amino acids. So this is not the end stage of digestion yet. Okay, this is not the end product yet of digestion, but it is the first stage in the digestion of proteins. Then the parietal cells secrete hydrochloric acid. Okay, so the cells that secrete the enzyme pepsinogen or pepsin is different. Pepsinogen is different from the cells that secrete hydrochloric acid. Okay, so this hydrochloric acid functions to kill the bacteria in food and prepare an acidic medium with an optimum pH of 1.5 to 2 for the faster action of pepsin. Now, thirdly, hydrochloric acid also stops the action of amylase, which requires a neutral medium with a pH of around 7 for it to function. So since hydrochloric acid is acidic, it will stop the action of amylase, right? Now, for mucous cells, of course, they secrete mucus. So mucus is a sticky, thick type of fluid, all right, that will coat the stomach wall, the inner lining of the stomach wall. And this protects the stomach wall from damage by hydrochloric acid and by the enzymes. Okay, because the hydrochloric acid is quite corrosive. It is a strong acid at a pH of 1.5 to 2.5. So we need to have the mucus to line the stomach, the inner surface of the stomach wall, and protect the stomach wall from damage by hydrochloric acid and also the enzyme like pepsin. So let's look at the structure of a gastric gland. How does a gastric gland look like? Now, so first of all, let's discuss the location of the gastric glands in the stomach. So if you cut open the stomach and we look at the inner surface of the stomach, the lining of the stomach, this is where the epithelial cells are found on the surface of the stomach. Okay, and this is where the gastric gland is found. So the epithelial cells will form the gastric gland. So in the lining of the stomach, there are gastric pits, something like depressions, okay, in the stomach lining. There are many of these gastric pits, and the cells that line the gastric pit forms the gastric gland. Okay, so if we just focus on one U-shaped structure here, this is a layer of epithelial cells, and they form the gastric gland. So the gastric gland is just a tissue, specialized tissue, made up of epithelial cells, okay, that are specialized to secrete substances. So the three epithelial cells in the gastric gland here, that's U-shaped, are the chief cells, the parietal cells, and mucous cells. Chief cells secrete the enzyme pepsinogen. Okay, so when pepsinogen comes in contact with the hydrochloric acid, it will be activated and it will be converted to pepsin. Then pepsin will digest the proteins to form polypeptides. Now, parietal cells in the gastric glands secrete hydrochloric acid to kill bacteria and to prepare an acidic medium for the efficient reaction of pepsin. Okay, then mucous cells produce mucus, so there are lots of mucous cells here, and the mucus has a protective effect to protect the cells in the stomach from damage by hydrochloric acid and pepsin. Okay, so normally the chief cells are found further down at the base of the gland, and then we have the mucous cells and the parietal cells at the sides here and at the top. So here are the notes about pepsin. Now, pepsinogen is an inactive enzyme secreted from the chief cells. So it's activated by the hydrochloric acid to become pepsin, and then the pepsin will hydrolyze proteins into polypeptides. So take note of this equation, which is the hydrolysis of proteins. Okay, So protein plus water catalyzed, this reaction is catalyzed by pepsin, and it will produce polypeptides. Now, what is the importance of hydrochloric acid in the stomach to humans? This is a past year SPM question, right? So if it's 3 marks, you have to try to give 3 different functions. Now, the functions or the importance of hydrochloric acid are to prepare an acidic medium with the optimum pH of 1.5 to 2 for pepsin to act efficiently at a high rate. So mention that at this optimum pH, pepsin acts efficiently at a high rate of reaction. Second function is to kill the bacteria in the food. Now, third function is to activate the pepsinogen, which is the inactive form of pepsin. So hydrochloric acid will activate pepsinogen to form pepsin, right? Then the pepsin can hydrolyze the proteins. And one more action is that it stops the enzymatic action of salivary amylase, because it will create an acidic environment that is not favorable for the salivary amylase to act, as salivary amylase needs a neutral medium. So here's a question concerning digestion in the stomach. What about renin? Is renin produced in human stomachs? Now, so renin is not mentioned in your new KSSM textbook, but it has always been mentioned in the older syllabus, okay, in the old textbooks. Now, first of all, let's find out what renin does. Renin is an enzyme that curdles milk or coagulates milk by converting the soluble casinogen into insoluble casein in the milk. So it changes the condition of the milk from a liquid state to a more solid state. Renin is definitely found in animals such as the cows or the young of the cows, the calves. And the benefit of curdling the milk is actually to increase the time in which the milk is retained in the stomach. In a more solid condition, it's easily retained in the stomach, so that there's more digestion of milk. Because if it's in liquid form, it can easily flow out from the stomach, right? So that's the benefit of renin in curdling the milk, so that it can be retained in the stomach, so that there's more digestion of the milk to produce more nutrients. Now, here is a reliable source that says that in animals that lack renin, for example, humans, milk is coagulated by the action of pepsin. So that's why many books do not mention renin as a human enzyme found in the stomach. Okay, so we humans can coagulate milk using the pepsin, according to this source. And human adults may not have renin, all right? But according to some sources, human infants do produce renin, except that the production decreases as the infant or baby grows older. Okay, so in the exam, if you're asked to give one example of an enzyme in the stomach, please do not mention renin, because I'm not sure whether the renin will be the one choice. If you're asked to give only one example, choose pepsin as the enzyme that is produced in the stomach to digest the proteins into polypeptides. That is a sure correct answer. Okay, so renin is maybe the secondary enzyme, okay, that you can write down if you're writing longer answers in the essay section. So digestion in the stomach. Now question, explain why hydrochloric acid and enzymes do not corrode the stomach lining. So we know that hydrochloric acid, the pH of 1.5 to 2 is very corrosive. Why doesn't it corrode the stomach lining? So the answer is there is a layer of mucus secreted by the mucous cells that protects the stomach wall from the damaging effect of hydrochloric acid and the digestive enzymes, such as pepsin. Okay, so the layer of mucus is a slimy protective layer that can prevent the corrosion by the hydrochloric acid and also enzymes. Number 7, food in the stomach is mixed with gastric juice containing hydrochloric acid and pepsin, and the food is churned by the peristaltic action of the stomach wall muscles for a few hours. So digestion in the stomach after a good full meal a big meal, can last 3 to 4 hours, where the food is, in which the food is being churned. Okay, it's the food is churned in the stomach. You know, something like when you look at the washing machine, it's churning the clothes. Okay, turning the clothes around. Okay, so the stomach can also churn the food, but of course not so vigorously like a washing machine. It does it by the peristaltic action, the contraction and relaxation of the stomach muscles. Then the contents in the stomach will slowly change after the digestion to form a semi-fluid called chyme. Then this chyme enters the duodenum when the sphincter muscle relaxes. The sphincter muscle at the bottom of the stomach will then relax. When it relaxes, it will open the opening that will allow the food to move into the duodenum. Let's go on to the next site of digestion, which is the duodenum. So the chyme from the stomach is released into the duodenum, which is the first part of the small intestine. Now, the duodenum doesn't produce any digestive enzymes by itself. So it gets the digestive enzymes from the pancreas. So pancreas secretes juice, pancreatic juice into the duodenum. And this pancreatic juice contains 3 enzymes, which have the short form LAT, L-A-T, the acronym LAT, which stands for lipase, amylase, and trypsin. Now, lipase hydrolyzes lipids to form fatty acids and glycerol. Amylase hydrolyzes starch to form maltose. Trypsin hydrolyzes polypeptides to form peptides. So trypsin continues the digestion. Okay, after pepsin has digested protein to form polypeptides, trypsin will digest the polypeptides to form peptides. Now, another organ that helps in the digestive process in duodenum is the liver. So it's another accessory organ, right? So liver produces bile, which is stored in the gallbladder, right? And this bile has a pH of 7.6 to 8.6. So it creates an alkaline condition in the duodenum, which is an optimum pH for faster action of these enzymes, lipase, amylase, and trypsin. Now, bile has another important function, that is to emulsify lipids by breaking down the lipids into smaller droplets to increase the total surface area so that the enzyme lipase can act more efficiently on the lipids. So bile doesn't contain any enzyme, right? It's just to emulsify the lipids. Okay, so digestion of carbohydrates, proteins, and lipids occur in the duodenum. All 3 classes of food are digested in the duodenum, which is the first part of the small intestine. So the small intestine consists of 3 parts: duodenum, jejunum, and ileum. Now, duodenum is the first part of the small intestine, which receives 3 substances: chyme from the stomach, bile produced by the liver and secreted by the gallbladder into the duodenum. It's a greenish color liquid. And then pancreatic juice secreted by the pancreas into the duodenum. Right, so this picture shows the organs involved in digestion in the duodenum, which is the first part of the small intestine. So the duodenum is connected to 3 accessory organs that help it to carry out digestion. Firstly, the liver produces bile, which emulsifies lipids and breaks down the lipids into smaller droplets. Next, we have the gallbladder that stores the bile. So gallbladder will secrete the bile into the bile duct and into the duodenum. It will flow into the duodenum when there is food in the duodenum. Next, another organ that helps in the process of digestion in the duodenum is the pancreas. The pancreas produces 3 important enzymes, which are lipase, amylase, and trypsin, to digest lipids, starch, and polypeptides, respectively. Next is the duodenum, which is the site for digestion of the starch, polypeptides, and the lipids. So it receives bile from the gallbladder and pancreatic juice via the pancreatic duct from the pancreas. Now, number 3, digestion in the duodenum. First, we're going to talk about the accessory organs that are not part of the digestive tract. Okay, they are not involved directly in the digestive tract, but they help in the process of digestion. Okay, so these are the accessory organs. The first one is the liver, and then you have the gallbladder and also the pancreas. Now the liver produces bile, which is a greenish fluid that is involved in lipid digestion. Whereas the gallbladder here, under this part of the liver, stores the bile. And then the bile flows into the duodenum through the bile duct here. So the bile duct is connected to the duodenum. Now, a common question asked is state 3 functions of bile. 3 marks. So answer, bile is used to first neutralize the acidic chyme that comes from the stomach. Secondly, prepare an alkaline condition of pH 7.6 to 8.6 for enzyme action in the duodenum. So the bile is alkaline in nature, and it prepares an alkaline condition so that enzymes from the pancreas, like lipase, amylase, and trypsin, will be able to react and digest the compounds or the food, right? Because the alkaline condition is the optimum pH for these enzymes to act. So bile prepares the alkaline condition. Now, lastly, a very important function of bile is to emulsify lipids by breaking down lipids, large lumps or drops of lipids, into tinier droplets to increase the surface area for digestion by the enzyme lipase. So bile doesn't contain the enzyme lipase, doesn't contain any enzyme. Its main function is just to break down the large drops or lumps of lipids to form smaller droplets of lipids. In this way, the total surface area of the droplets increases. Okay, is larger. The total surface area of the drops, small drops, are larger than that of one big drop. So after that, then the enzyme lipase can act efficiently on the surface of these drops of lipid to break down the lipids to form fatty acids and glycerol. Okay, now where does the enzyme lipase come from? It comes from the pancreatic juice. Okay, whereas bile is from the liver. So if you're asked to give the functions or state the functions of bile, just one function, please choose please choose C as the most important function. Okay, and then B, and then only if there are 3 functions, then you can write in A. Now the second accessory organ other than the liver that helps the duodenum to carry out the process of digestion is the pancreas. So the pancreas is a leaf-shaped organ that's connected to the duodenum, and it secretes 3 enzymes through the pancreatic duct into the duodenum. So these enzymes are very important. They are lipase, pancreatic amylase, and trypsin. Think of L for lipase, A for amylase, and T for trypsin. LAT. Okay, it's like a name of a person. So what are the functions of these 3 enzymes? So let's look at the site of the digestion. Site of digestion is the duodenum, into which the enzymes LAT have been released from the pancreas into the duodenum. So the first enzyme is amylase. Pancreatic amylase hydrolyzes starch to maltose, and the word equation is starch plus water. By the action of pancreatic amylase, okay, produces maltose. So trypsin, on the other hand, hydrolyzes polypeptides into shorter peptides. So the protein molecule is slowly broken up, right? Because it's a complex molecule. So pepsin has already hydrolyzed proteins to polypeptides. Now trypsin hydrolyzes the polypeptides into shorter peptides. Okay, so the equation is polypeptide plus water catalyzed by trypsin. We have the formation of peptides. Okay, lipase hydrolyzes lipids into fatty acids and glycerol. So equation is lipids plus water. With the action of lipase, the enzyme lipase, fatty acids and glycerol are then produced. So try and memorize the 3 enzymes from pancreas and also the equations for their reactions. So now we come to the fourth site of digestion, which is the ileum, is the last part of the small intestine. So ileum has intestinal glands that secrete enzymes, unlike the duodenum that does not secrete any enzymes, right? So ileum has intestinal glands to secrete intestinal juice that contains 5 enzymes, which has the acronym SMELL, S-M-E-L-L. So the first enzyme is sucrase. Sucrase hydrolyzes sucrose into glucose and fructose, whereas maltase hydrolyzes maltose to form glucose. Lactase hydrolyzes lactose to form glucose and galactose. Now, all these 3 substrates are actually disaccharides, and they are broken down to form the monosaccharides, which are the final products of digestion. Okay, so the final products of digestion of carbohydrates are glucose, fructose, and galactose, the monosaccharides. And also take note that the name of the enzyme and its corresponding substrate is easy to connect or relate to. Okay, now if we have sucrose, the enzyme must be sucrase. Okay, just change the OSE in the substrate to ASE in order to get the name of the enzyme, right? So other than the digestion of disaccharides, we also have the digestion of peptides. So erepsin hydrolyzes peptides to form amino acids and thus completes the digestion of proteins. So the digestion of proteins begins with the enzyme pepsin in the stomach, where proteins are digested to polypeptides. And then in the duodenum, trypsin hydrolyzes polypeptides to peptides. So peptides are then hydrolyzed by erepsin to form amino acids, which are the final products of digestion of proteins. And then there is again lipase, as in the duodenum. So this lipase is secreted by the intestinal juice, and it hydrolyzes the lipids in the ileum to form fatty acids and glycerol. Okay, so the digestion of lipids is only one step where the enzymes are concerned. Okay, lipids are digested straight away to form fatty acids and glycerol. All right. So the fourth site of digestion and the final site of digestion is ileum, and these are the long notes for it. Based on what we have discussed in the table, is almost the same, right? So glands on the ileum wall secrete mucus and intestinal juice that contains 5 enzymes: sucrase, maltase, lactase, lipase, and erepsin. Acronym is SMELL. The alkaline medium in the ileum is at the optimum pH of 7 to 8, right? So it's alkaline, and it allows the enzymes to act efficiently at a high rate of reaction. So please take note that in the mouth, the pH or the condition is neutral. Then in the stomach, the condition is acidic with a pH of 1.5 to 2. But in the small intestines, that means the duodenum and the ileum, the condition there is alkaline, right? The medium there is alkaline with a pH of, for example, in the ileum is pH of 7 to 8. So the ileum is the lower part of the small intestine here. Okay. And in the last part here is to again explain the digestion of food in the ileum, right? Which uses the enzymes SMELL, the 5 enzymes that are secreted by the intestinal glands, okay, for digestion of food in ileum. So we have already discussed this in the table just now, so I'll go through very quickly. Sucrase hydrolyzes sucrose into glucose and fructose. The equation is sucrose plus water, sucrase, with the action of sucrase, glucose and fructose is produced. Now, always remember to write the water, okay, water as a substrate in your hydrolysis reactions, okay, because it's a hydrolysis reaction, so water is involved as a substrate and it reacts with the food. Now, maltase hydrolyzes maltose into glucose. So maltose plus water, with the action of maltase, glucose is produced. Lactase hydrolyzes lactose into glucose and galactose. So lactose plus water, with the action of lactase, produces glucose plus galactose. Lipase hydrolyzes lipids into fatty acids and glycerol. So lipids plus water, with the action of lipase, fatty acids plus glycerol are produced. Erepsin hydrolyzes peptides into amino acids. So peptides plus water, through the action of erepsin, forms the amino acids. Now lastly, questions are usually asked about the digestion of carbohydrates in the body, or digestion of protein or lipids in the body. For example, you can be given a question like this. A student eats a bowl of white rice. Explain the digestion of white rice in his body, right? So white rice is starch, mainly starch. So we have to know all about the digestion of starch in the human body. Where does the digestion of starch start? Okay, it starts in the mouth. So we have to mention that amylase digests or hydrolyzes the starch to form maltose in the mouth, okay? And mention all the glands and secretions. And then in the duodenum too, there's digestion of starch by amylase, right? To form maltose also. And this amylase is the pancreatic amylase, right? Whereas the amylase in the mouth is salivary amylase. And then lastly, after maltose is formed, that's not the end of digestion, the digestion process. The maltose is digested by maltase in the ileum to form glucose, right? Okay, so that's an example of a question that can be asked. Therefore, we have to know all about the digestion of carbohydrates in the various parts of the digestive system, all right? So for carbohydrates, digestion of carbohydrates occur in the mouth, where starch is digested to form maltose by amylase, and also in the duodenum, there's digestion of starch into maltose by pancreatic amylase. And finally, in the small intestine, the ileum, in the ileum, there's digestion of the disaccharides to form the monosaccharides. Okay, so that's the digestion of carbohydrates, right? So you must know all the enzymes like amylase, sucrase, maltase, lactase. Then for digestion of proteins is in pink. Okay, so digestion of carbohydrates are all in yellow. You can highlight that in your notes if you have copied the diagram or the table. And for digestion of proteins, we can use pink color. Okay, so digestion of protein starts in the stomach when pepsin digests the proteins to become polypeptides. Okay, you can mention renin also if you want to. So renin curdles the milk and converts casinogen, which is soluble, into insoluble casein. Then in the duodenum, trypsin continues the digestion of protein, whereby polypeptides are converted to peptides. Okay, you can take note here. Pepsin produces peptides, right? Pepsin hydrolyzes proteins to form polypeptides, but trypsin hydrolyzes polypeptides to form peptides. And erepsin then continues the digestion of the peptides. Erepsin hydrolyzes peptides to form amino acids, right? So digestion of protein occurs in the stomach, the duodenum, and the ileum. Okay. Now, how about lipids? For lipids, lipase in the duodenum digests the lipids into fatty acids and glycerol. So you can highlight the digestion of lipase using another color like blue, right? Or green. Then there is also the production of bile to emulsify the lipids by breaking down large lumps of lipids into smaller droplets for the digestion, faster digestion by the enzyme lipase. And lastly, in the ileum, there's also the secretion of lipase that helps to hydrolyze the lipids further, any remaining lipids or some of the remaining lipids into fatty acids and glycerol. Okay, so digestion of lipids occur in the duodenum and the ileum, whereas digestion of carbohydrates occur in the mouth, but not in the stomach, and then the duodenum and the ileum. Okay, so this table is very useful as it shows you the digestion of the different classes of food such as carbohydrate, proteins, and lipids. by highlighting the various classes of food with the different colors. Okay, so you can also do that too for your table. And so this table will be very helpful to you when you do your revision. Now, as a quick summary here, for Chapter 9 overview on the various processes involving digestion and movement of nutrients in the body, we have this chart here, which is also quite useful. So the processes involved in nutrition are ingestion of food, that means eating the food, putting it into the mouth, where there is the enzyme amylase from salivary glands to digest the starch. And then going on to the stomach, the enzyme pepsin is released to digest proteins. Okay, so enzyme pepsin from gastric glands. Then going down to the duodenum, bile is secreted from gallbladder for emulsification of fats. And enzymes lipase, amylase, and trypsin, or LAT, are secreted from the pancreas to digest the various food substances, okay, such as lipids, starch, and also polypeptides. Polypeptides are digested by trypsin. Next, going down to the ileum, enzymes sucrase, maltase, lactase, epsilon, and lipase, or SMELL, smell, are secreted from the intestinal glands to digest the disaccharides, which are sucrose, maltose, and lactose, and then to also digest, epsilon digests the peptides to form the amino acids, and lipase digests the lipids to form the fatty acids and glycerol. So in the ileum, the end products of digestion are formed, okay, such as glucose, fructose, galactose, and also amino acids and fatty acids and glycerol. So after that, the food has to be absorbed. So there's absorption of digested food into the blood, capillary, and lacteal in the villus of the ileum, so that the simple products of digestion of food, simple products of food digestion, will go into the blood and is transported to the liver, okay? And then the liver carries out a process called assimilation and also sends some products to body cells where assimilation continues, where products are used to build more complex substances for the body's needs, and so forth. So we are at this stage now. We have finished digestion, and in the next few videos, we'll continue with absorption of digested food and then with assimilation. Right, so now, as a bonus, I'm going to give you a super acronym for the 10 digestive enzymes in the human body, so that you can remember all the 10 digestive enzymes that we have discussed in this video. The acronym is AMY, peeved by lut smell. AMY is peeved or turned off by or dislikes the smell, okay, from lut, because lut may have just sweated a lot, or he didn't take a bath, or he had body odor, right? So AMY is peeved or put off by lut smell. So what are the 10 enzymes in the right order, right? We start with AMY. AMY stands for what? Yes, amylase. Peeved, the PE stands for the enzyme in the stomach called, yes, pepsin. LAT stands for the 3 enzymes from the pancreas, which are, yes, lipase, amylase, and trypsin, right? And SMELL, the smell, stands for the enzymes in the small intestine in the ileum, right? So remember, intestines smell, okay? So what does SMELL stand for? Yes, sucrase, maltase, epsilon, lactase, and lipase. All right, so it's already in the right order. All these 10 enzymes are in the right order, starting from the mouth to the ileum, right? So amylase is in the mouth, pepsin is found in the stomach, or X in the stomach, and LAT, the 3 enzymes from pancreas, are secreted into the duodenum to digest food. Finally, enzymes SMELL, smell, are found in the ileum to complete the process of digestion. Okay. Now, for some of you, you may want to include renin as one of the enzymes in the stomach. So that's an alternative for you. AMY, repelled by lut smell. AMY is repelled by lut smell. The word repel here has a similar meaning to being peeved or turn off by the smell, right? To dislike the smell. So RE stands for renin and PE stands for pepsin. Okay, so this is an alternative acronym. So it's up to you whether you want to include renin or not, right? Textbook doesn't include renin, but maybe some teachers or in some cases, the marking scheme may include renin as one of the enzymes in the stomach, okay, based on the old syllabus. So never mind, you can use Amy repelled by large smell if you want to, okay? Right. So that's all for this lesson. I hope you have enjoyed the lesson and also benefited from it. Please do share, like, and subscribe. And thanks for viewing. So take care for now. Till we meet again, goodbye.